This is part 4, the nervous system, specifically the central nervous system. We could talk about this in class. Please bring this person up. This is kind of making a segue from what we've been focusing on, looking more at cell communication and chemical signaling as opposed to the integration portion. But do not forget this process. We've talked about this division. Make sure you know it. We have two types of matter in our brain. We have our gray matter and we have our white matter. There's also ventricles, which are space. Take a minute to look up the difference between gray and white matter, because we will address this question when I see you in class. The central canal of the spinal cord and the ventricles of the brain, which are hollow, are filled with this stuff called cerebral spinal fluid. And this spinal fluid basically is filtered from the blood, and it protects the brain, it provides a cushion, and it provides nutrients and waste because it's kind of cycling through these cavities all the time. This is why if you ever get in a car accident or someone has a very serious head or traumatic head injury, having blood come out of their ears, obviously bad. If we see clear fluid leaking out, it means that the blood-brain barrier has been broken and that cerebral spinal fluid is leaking out, which is actually far more dangerous than just blood. Blood could indicate any sort of inner ear damage, whereas cerebral spinal fluid leaking is very bad. In the central nervous system, we have these cells called glia, and glia have numerous functions. There's multiple types, and their job is to nourish, support, and regulate the neurons that are actually conducting impulses. We see that certain types called embryonic radial glia form tracts along which newly formed neurons migrate, so they allow for neurons to move to where they need to be. Astrocytes induce cells lining capillaries in the CNS to form tight junctions, resulting in the blood-brain barrier. So astrocytes basically create this protection wall, but in both cases, neither of them are actually conducting a message. When we look at the central nervous system and the different divisions that are going on, we want to look at all the different types of cells and what happens during information processing. Anything in the dark orange are peripheral nerves and part of the peripheral nervous system, so obviously that's very complex. The central nervous system, all the processing occurs in the spinal cord and in the brain. In this yellow image, or the yellow wheel, basically. As we saw before, the vertebrate brain is regionally specialized. Basically, that means that as we've evolved, our neurons have started to accumulate and amass in certain places, which means we end up getting a brain and a vertebrae. And then to go even a step further, we see that specific brain structures are specialized for certain functions. So not only do I have a mass of brain cells that process information, there's actually certain parts of my brain that process certain parts of the information. These structures arise during embryonic development. When we're talking about human brain development, during 1 month gestation, not a lot of brain development happening. It is just mostly soft tissue. Once we get to moving past 1 month, this is when the nervous tissue starts to kind of differentiate or become separated parts. Post 1 month, we see it basically has 3 main regions: the forebrain, midbrain, hindbrain, and that's about it. Once we get to 5 weeks, so almost a week, not only a week after, into month 2, we see that the division of nerves into specific categories increases very quickly. Also, the image above will show you what those parts end up developing into. And we'll talk about those adult parts. So, in an adult and also in a child, these are not fully formed, but the parts are all there. We see the cerebrum, which is yellow. This includes the cerebral cortex, white matter, and the basal nuclei. We have the diencephalon, which is orange, thalamus, hypothalamus, epithalamus. We have our midbrain, which is purple, part of the brainstem, our pons, also part of the brainstem, and our cerebellum, and we also have our medulla oblongata. These are all kind of the core regions of the brain that we see developing once a child is born. This is a view from the rear, just so we have an idea about that as well. And this is, again, kind of dividing it a little bit further. So we know that within the brainstem, we've got midbrain, pons, medulla oblongata. And the brainstem itself basically controls anything that is subconscious, that is innate, breathing, temperature regulation, anything that we don't actually think about but is happening all the time. Digestion, metabolism, all of those things happen in the brainstem. And the diencephalon, that also controls some of those things, but these are the regulatory organs of the brain. They produce chemical signals that in turn control how fast is my heart beating, how quickly is my metabolism moving. Our cortex, or the kind of sensory processing area, is divided into lobes. We've got our frontal lobe, which is decision-making and planning. We have the very back of the frontal lobe, which is the motor cortex, which actually controls our movements. This is why if someone is drinking or under the influence, it not only influences their decision-making, but also their motor control. It's because the frontal lobe is impacted completely. The somatosensory cortex, this is our sense of touch. It is part of the parietal lobe. The parietal lobe is sensory association cortex, basically taking all of our senses, smell, touch, sight, sound, and integrating them into our experience. The occipital lobe is going to be our visual association cortex. It takes all of the information from the optic nerves and combines those images and determines what we are actually recognizing. If it's something we've seen before, we can put a word to that image. Cerebellum, we'll talk about a little bit more. The temporal lobe deals with auditory hearing, essentially. Two interesting areas that we should know. Wernicke's area is comprehending language. Broca's area is forming speech. And so we sometimes see if people get in a car accident or people have these damaged areas, they either cannot form speech or they can't understand speech. And this is really interesting that we found like very specific areas in the brain that control our ability to speak and be spoken to, or ability to understand what is being said to us. And we can talk about this a little bit more in class. Please feel free to bring it up. The cerebral cortex receives all sorts of input from the sensory organs and from the somatosensory receptors. Somatosensory receptors provide information about touch, pain, pressure, temperature, how I'm sitting, if I'm walking, if I'm running. And the thalamus directs this different types of input into very distinct locations. Another thing to think about is that each of the regions of my cortex have a very specific designation of what they do. This shows where the input, the touch input from all of these different areas goes very specifically on the brain. This person is actually called the homunculus. It also shows the amount of nerves based on the size. So we can see that our tongue, our face, our hands, and our feet actually have the largest concentration of nerves. These are the most sensitive. Again, we can see it here too. The frontal lobe basically controls decision-making and emotional response. These are not developed until we are in our mid to late 20s. It basically impacts our executive functioning skills, our ability to make rational decisions, taking into account all of the information that we are seeing.